Alright, welcome back friends. In this video tutorial, we'll be talking about lentiviral gene therapy. Now we have seen what is gene therapy and what are the implications of gene therapy in future, and we have also seen how we can utilize adenovirus for conducting gene therapy. Now let us see another type of virus. It is called the lentivirus, which is in the family of retrovirus. So let us look at how we can utilize this retroviral uh, member, which is the lentivirus, for the gene therapy purpose. Now before going into the further discussion, I must tell you first basic thing. Assuming that you all know about what is retrovirus and what is a process of retroviral infection, I simply want to tell you is that retrovirus is a very good advantageous virus for the gene therapy, because the normal retroviral life cycle involves in the insertion of the retroviral genome inside the host cell DNA, right? Because suppose it is a retrovirus. Now this retrovirus is containing its own DNA, and now let's say this is a host cell. So let us conduct this is a host cell. When the virus enters into this host cell, what it does, inside the host cell there is host cell genome. For example, say this red color thing is a host cell genome. Say this is the host cell genome. Now inside somewhere there in the inside this host cell genome, after the insertion, it enters its own DNA or genetic material. Say this blue colored region, right? So once after invading the host cell, it inserts its own genetic material inside the host cell. Here we can see it, and this procedure is really important, and this is unique feature about the retrovirus, right? Because the retrovirus means if they are simply utilizing reverse transcriptase enzyme to produce the DNA from the existing single stranded DNA or single stranded RNA, and they will produce the DNA, and then the cDNA they will incorporate that DNA onto the host cell genome, and then the host cell genome will be replicated and multiplied so that they produce more and more copies of their own. Okay, so for this attachment they want another protein which is called integrase, and also there are other proteins for the cleaving and cutting this segment out for the packaging purpose. So those these retroviral genes are having three different types of genes. Actually, one is their genomic DNA, second one genomic DNA or RNA, whatever, which is their genome. Then the envelope protein coding segments, because the envelope segment that they are having are very pathogenic for us, right? So please try to maintain this. So there are actually they are having three different major genes. One is GAG, which are coding for structural part or other structural part or structural regions, except for the envelope. Then Pol, which is coding polymerase, which is helping them to replicate their genome inside the host cell. And thirdly, it is ENV or envelope. Or ENV is called is a gene which is coding the protein to produce the envelope region, right? So these three are the major type of genes that are found inside a retroviral genome. Now inside lentivirus also we are going to see the expression of these three genes, but except for these three genes there are also many genes like TAT, like REV, and many more genes, and we are going to see their functionality. Now again, the gene therapy means it's all about modification of an existing host in such a way so that we can utilize it in the beneficial purpose in the gene therapy. Now the existing wild type virus is lentivirus. We cannot use it, we must modify this. So we must take those part out, we must cut those part out which are responsible for the pathogenicity. For example, this ENV part, so we must cut this part out. But if we cut uh, so so if we cut this part out, so we need to supply this envelope via another procedure, via any another vector. So we need to rely upon another helper vector, and that is the procedure of the whole gene therapy. When we modify our wild type, which is providing us the gene of interest, or which is providing us the region for inserting our desired gene, we may what we can say, we sacrifice several different gene products, and for that purpose, for the establishment of for the establishment of the infection and the release of the virus particles, we must have provided all of these segments inside the host cell by co-transfecting some complementary cell line with some other helper vectors. That's the actual goal of most of the gene therapy, and we have also seen in case of adenoviral gene therapy too. Now in this procedure I'm also going to tell you the adenoviral gene therapy examples, so I recommend you to go back and first look at the adenoviral gene therapy, then come here. So the lentiviral vector development purpose, let's first talk the basics, as this lentivirus is also a type of retrovirus, but it is much more complex than some oncogenic retrovirus like HIV. Usually what they are having, as I've told you before, they are having gap precursor, pol precursor, envelope precursor mainly. Envelope precursor provides this envelope structural proteins which are extracellular and can be intracellular. The pol precursor produce polymerase, reverse transcriptase enzyme, which is in this case acting as a polymerase. Integrase, which help them to integrate its own genome inside the host genome, that I have told before, and protease, which is required for further cleavage of its own DNA sequence and to package it. Okay, and also the GAC precursor producing the core structural protein, which is helping them to produce the structural regions or structural part of this particular virus, because the viral not only needs its DNA or genetic sequence, but also it needs its structural sequences to be inserted, to the genes to be inserted. Okay, now here uh, the significance of lentiviral vector development are viral components that enable HIV productivity to infect non-dividing and terminally differentiated cells. That is the advantage. Now the lentivirus are capable of transducing quiescent cell. This is another very important assumption. Not is the assumption, is actually proven fact. Now the process is facilitated by several different parts. So first is the integrase protein, then the matrix protein, then VRP, VPR, and PPT. So these are the proteins which help them to attack the quiescent cell and establish its own what you can say, its own placement inside this host cell, right? So we don't want to go to the basics and detail about how they integrate inside the cell on all these things, because I've told you before, because they utilize their this integrase protein, matrix protein, to attach with this host cell and evade inside the host cell. Once they are attached and evade inside the host cell, they start releasing its DNA genetic material, and its genetic material is inserted or incorporated in between somewhere middle or somewhere inside the host genome, and using the integrase enzyme. Once the process is established, when everything is established, then they take up and cleave their own genome segment out of that host genome part, then utilizing this kind of protease enzymes. Okay, now let us look at the constituents of making the gene therapy vector. Now for the making of this kind of vector utilizing lentivirus, we again require three different type of elements. In case of adenovirus, we have seen three elements. In case of here also, we need three elements. Definitely one element we must provide us the gene of interest. So here it is a transfer plasmid which carries our gene of interest. Now this plasmid is again a modified lentiviral vector. We also need a packaging plasmid which will provide us the ENV segment, because if we give the ENV segment in this transfer plasmid, what it will do, it will act or behave like a wild type virus, because the ENV is pathogenic, so it can infect the cell, it can kill our host cell. We don't want this, so we must supply ENV using another vector. Here it is supplied using a packaging plasmid, and transfer plasmid is also only carrying a gene of our interest, and also it is having some segment, some few segment or few gene segment of our host cell. And third is of our not host cell, of our lentivirus, sorry. Third is an envelope encoding plasmid. Also the envelope encoding plasmid is really important, and a packaging plasmid important, which will provide us all the structural parts, right? So I made a mistake before. So actually packaging plasmid is providing, let me change the color. Packaging plasmid is providing us all the structural parts, which will give us all the structural necessary, inside which we package our DNA or genetic material. Transfer plasmid is providing us the gene of interest. So now gene of interest is there, structure is already made. And third is an envelope encoding plasmid, which will provide us the envelope, because the envelope is important in this case. Now remember, in case of adenovirus, envelope is not required because the protein coat is acting as the terminal barrier. But in this case, there is an envelope outside this protein coat, so we must have this envelope, because this envelope is important to establish the infection inside the host cell. So that's why the envelope is important, and we must have access to the envelope, and the envelope is encoded and provided by an envelope encoding plasmid. So you must have three plasmids. One simply provide only envelope, one provides only structural proteins, and another provide the gene of interest. Okay. So usually HIV derives the virulence from 9 genes. So the virulence factors that are present in HIV are from 9 genes. ENV is one of them. VIF, VPR, VPU, NF, and TAT. Now what we need to do, we must have deleted or eliminated as more as of these 9 genes, right? Because these genes are pathogenic, it can create problem for our host cell, it can kill our host cell. So we must have released or must have cleaved these particular parts out. So for that we must have taken this one out, we take this, all this, most of them out. Now what with this, out of these 9 genes, 6 can be removed without altering the HIV's gene transfer ability, as we can see here. So from this 9, if we delete 6 known genes from here, it won't affect the HSV gene transfer ability. So what we can do, uh, we can drag out at least or maximum 6 genes, not more than 6 in this case. Okay, so we can eliminate 6 1 and 3 genes will be present, so the level of pathogenicity will be less. So we'll do this, we'll take out those 6 genes which can be just cleaved out. If we cleave those rest 3 out, then we need to compromise some facts because those genes are required for the gene transferability. So in those cases we must provide some other factors from outside so that the gene transferability is not get hampered, right? So here it is the construction of the 3 types of plasmids. As we can see, this is a whole wild type lentiviral genome. Now if we provide this whole wild type lentiviral genome, it can create problem, it can create infection, so we won't provide this. Instead we provide this in 3 different fragments using a co-transfection experiment. One is only with envelope protein or envelope plasmid, envelope protein encoding plasmid, which is the third kind. Another one is simple transfer plasmid, which will carry our gene of interest, and it will have our LTR at both the ends, just like the structure of the adenoviral uh, plasmid. And the third one is the packaging plasmid, because it will have the GAG, POL, TAT, RAV, these sequences, because these GAG, POL sequences are required for the production of protein encoding segments or the structural components of the viral particle. Okay. Now the generation of gene therapy vector using this lentivirus or HIV virus. Now as we have seen, there are several segments, so we need to prepare 3 different type of vectors, and we'll use these 3 vector to co-transfect the host cells so that they can work in complementarity with each other, right? But we cannot provide everything in a single genome, right? Now here in the first generation, it is composed of HE minus the simple envelope protein uh, at the very beginning and the first generation of plasmids. But the second generation of gene therapy, 5 of the 9 genes are deleted, only GAG and POL or TAT and RAVE genes are retained. Okay, and it seems to be very active, very good. Now in third generation it is further modified and it contains only GAG, POL and RAVE gene instead of lost of this 9 genes. So we have deleted 6 genes and we kept only 3 genes, which are the GAG, POL and RAVE. Now GAG is providing us the structural components, POL is again providing us the enzymatic components like the protease, like the integrase, which are required for the insertion deletion purposes. And RAVE gene is also there. Now the RAVE gene is again helping us in the establishment of our infection. Okay, now here it utilizes a chimeric 5' LTR to ensure the transcription in the absence of tag. Now here it suggests us that LTR is playing important role in the transcription purpose, because this LTR is acting like the factors or transcription factors to initiate the transcription or to stop the transcription process. Okay. Now this is a simple modification in the very beginning, simply deleting the genes that are previously present in the wild type lentiviral genome or wild type HIV gene. But in this case what we are seeing, further modification must be done. Simply what we can do here, so what we have seen in this previous case, we have these LTRs which were previously there, we have gene of our interest with promoter sequences. But what we'll do here, we will give the regulatory elements or WPRE, which is called Uchak hepatitis virus post transcriptional regulatory element or WPRE, to increase the level of transcripts. So we'll provide these WPRE elements downstream of our desired gene, right? We provided this is something which we add from outside, but rest of the part we have discussed in generation 1, generation 2 and generation 3, simply deleting the wild type genes that are present there, which can be pathogenic. Okay, now this is one modification. This is further modification that has been conducted. And also the transfer vector are designed as self inactivated. Now what we mean by self inactivating, now this is achieved by removing the U3 region. So you can see this is the LTR part is made up with U3, U5, and then the transcript start, which is R region. Now what we do here, we simply remove this U3 region of the 3' UTR. So you can see here in 3' LTR we don't have any U3 region. So is there any U3 region? The answer is no, there is no U3 region. So we deleted this U3 region from this 3' LTR, but not from 5' LTR, because 5' we are having this U3. Now by deleting this U3 at the 3' LTR region, it results in the virus being unable to reconstitute its promoter. So it cannot reconstitute its promoter. So what we are ensuring, we ensure that whatever gene we are providing, it's under the control of promoter, and it is maximum control under promoter. Now if this U3 region is there, it was seen that this U3 can help this particular viral vector to reconstitute its promoter, and the expression of gene can be altered. So for preventing that, we make this self inactivating so that it cannot be inactivated again or reactivated again by itself, right? And also you have the size side for the packaging signal, right? So we are having all these important parts inside it. Okay, now what are the difference between the second generation and third generation vectors that we've talked about? The second generation vector is can be packaged only by a second generation packaging system, but in third generation vector can be packaged by both second generation and third generation packaging systems. Packaging systems means simply they are the different types of host cell. The packaging plasmid that are present in second generation are GAG, POL, RAVE, and TAT, but in third generation we come down to only three, which is GAG, POL, and RAVE. Now the envelope plasmid that are present in this case usually code for VSVG, right? But in third generation, the envelope plasmid is again code for VSVG, which is similar in both the case as we have seen. Okay, and also the promoter that we have seen is altered in the third generation vector, because we have seen that 3' LTR is modified, the U3 is deleted, which was not in second generation. And also safety measures, second generation vectors are safe because we must care about safety first. The second generation vectors are safe. It uses separate plasmids for the encoding of viral HIV genes. But in case of third generation, what we have done, instead of separate one or two different vectors, we are providing four different plasmids for our gene therapy purpose. So we are getting safer and safer transfer of the genetic problems, right? So this third generation vectors are way better than the second generation vectors. Let me change the color here. It is way better in many aspects than the second generation vector. So that's why it is third generation and new generation vector, right? Now the third generation in lentiviral vector, if we look at it, it's a packaging. This packaging system offers maximal biosafety. That's the very advantageous fact about this third generation vectors. But again, they are constituted with four different plasmids. Remember, in case of adenoviral vector, we have taught three different plasmids. We utilize three plasmids for construction of this um, whole disarmed viral vector, but in this case we utilize four different plasmids instead of three. Okay, majorly three plasmids. One is the transfer plasmid having our gene of interest. Sorry for the bad handwriting here. Gene of interest. In the packaging plasmids providing GAG-POL, which is the protein structural part, and envelope plasmids, which are giving us VSVG, or the envelope of this virus, which is helping them to coat this whole protein content. Okay, so we must provide these three using three different plasmids, but not by same. So we'll be looking each of these plasmids and at their details. Now here this first one is a packaging plasmid, consists of the GAG and POL regions. So you can see here GAG POL genes are placed here in this part, okay, which is providing us the proteins that are required for the production of structural constituents. And also what we are having ampicillin resistant part, and all of these vectors are designed in such a way that they are having the cloning regions or the restriction enzyme sites, as well as they must have some selectable marker regions. Here it is having the ampicillin resistant marker, so that we can get it or select it after the transformation onto the E. coli cell. Okay, Now in this case, expression of this GAG and Pol genes from PLP1, it is called the PLP1 or plasmid LP1, has been rendered a rep dependent process, right? Because except for the rev, this particular plasmid cannot be replicated, right? And they are modified in such a way so that in the absence of rev, they cannot be they cannot be replicated, right? So to ensure that, we must have add this RRE sequence somewhere there in this plasmid. Now this RRE sequence prevents the GAG and Pol expression in the absence of rev. So if there is no rev expression, then GAG and Pol expression will not be there, because the rev is important. Rev is providing the signal. Now it's the time for packaging, so the viral will start to package. But if there is no rev, there is no meaning for packaging, because the cell is not ready yet to package the viral particles and to release the particles outside, right? So once there is rev only, RRE is getting activated so that the GAG and Pol is getting expressed. But there is no rev, no RRE expression, no GAG Pol expression, so no structural proteins are made. So this is the basic process, basic uh, construction of the LP1 vector here. Now here are the different regions of this part, and the different type of benefits are also provided. Like here, beta globin intron is provided to enhance the expression of GAG and Pol gene. So this beta globin intron is provided for the enhancement of this expression, and RRE is provided for the regulation of the expression of GAG and Pol. Okay, and also we are having the origin of replication, which is the PAC origin, right? Because it permits high copy replication and maintenance this particular plasmid inside the E. coli cell. So it is perfectly designed for the maintenance of this plasmid inside the E. coli cell. And I've already told you about the ampicillin. And okay, so and also we are having the promoter for CMV. The promoter for CMV is cytomegalovirus, as we all know. It permits the high level expression of HIV1 GAG and Pol genes in mammalian cells. So that's why this promoter is chosen, because whenever we try to express some of our desired products inside the mammalian cell line, it's the best option to choose the CMV promoter, right? And most of the case, not only mammalian, but if we are want to express our desired genes or desired plasmid expression inside the eukaryotic cell lines, we must choose this CMV promoter. Okay, now this is the second one, which is the transfer plasmid containing our gene of interest, right? So here it is, cDNA is provided in this case. So transfer plasmid is having TUD and TUT. This plasmid is helping us for the transfer purpose, right? Now nowadays in the fourth generation plasmid, third generation plasmid, what they are providing, they provide these three segments, and this transfer plasmid no longer carries our gene of interest. Instead, here we can see, here you can see, so here it is, the LP2, it is designated as LP2 or lentiviral plasmid 2 here. It is having 4180 base pairs. This is not required, but what is required in this case is having this RSV enhancer, which permits the high level expression of this rev genes, right? Similarly, in this case of, as we have seen, this beta globin introns is there for the expression of GAG Pol. Similarly, here we are having RSV promoter for the expression of rev gene, and again HIV rev operon ORP is also there. It encodes the rev protein that interacts with the RRE segment of the previous APLP1 plasmid to induce the GAG Pol expression. Now here we can see the molecular crosstalking between these two plasmids simply using rev. Because remember, as we have seen, the replication of this GAG and Pol, the translation of GAG and Pol is simply rev dependent, right? The process is also rev dependent. So only once the rev protein is secreted and they interact with this RRE region, GAG and Pol will be expressed. So here, that is the important part. So this is a crosstalk between PLP1 and PLP2. Now here we are having also HIV promoter here. It allows the efficient transcription termination and the polyadenylation of the mRNA, because what we will be doing, we are trying to develop the RNA in such a way so that it can be inserted inside the host eukaryotic cell, because we need to incorporate into the host cell. That's why this HIV PA, which is unique here in this case, can be present there. Okay, and ampicillin resistance is there also to select this particular plasmid from a group of plasmid using ampicillin screening or antibiotic screening. And PAC origin is also there for high copy replication and maintenance of this particular plasmid inside the E. coli host. Okay, so this PAC origin is common for PLP1 and PLP2, and they all have this ampicillin segment, but what they are varying in this case, it is having rev, and the previous one is having GAG-POL. Now the rev will control the expression of GAG-POL. Okay, so these two, PLP1 and PLP2 plasmids, will help each other for the establishment of their infection. Okay, or establishment of the infection, we can say that. Okay, now the third part is the envelope plasmid, which carries only envelope encoding segment, which is here VSVG. This is the envelope encoding segment. Okay, so it encodes for the envelope G glycoprotein from a vesicular stomatitis virus, and that's why it's called the VSVG, vesicular stomatitis virus, and G stands for the G glycoprotein. Okay, envelope G glycoprotein. Okay, and this is a broad range host actually. Okay, it can attack broad range host. Now here also we are having beta globulin intron. Why we are having beta globulin intron? Because this beta globulin intron again allows the efficient transcription and termination of and also polyadenylation of the mRNA. So the task here fulfilled by HIV-1-PA, here it is conducted by beta globin. So beta globin PA, it is conducted by this one. Okay, okay. Now there is Park origin. Park origin is given, remember, the same purpose: permit the high copy replication and maintenance of this particular plasmid inside the E. coli host cell, and also ampicillin resistance marker for screening of this particular plasmid containing E. coli cell from a bunch of cells using the antibiotic screening method. And here it is the PCMB, promoter for cytomegalovirus, right? So this virus again permits the high level, this particular gene is again permits the high level expression of VSVG gene in mammalian cells. So everything is again provided here. It is called PLP or VSVG vector. It is slightly larger vector than PLP2, and a very important part is again the constitutive promoter, the constitutive promoter, which is simply called the RSV promoter. Now the promoter they provide here is RSV promoter, which is further modified. Somewhere here it should be RSV promoter. So the gene is from the VSV or vesicular stomatitis virus, but here is the promoter is from RSV. Okay, why? Because this promoter is pretty strong and it is a constitutive promoter. Providing a constitutive promoter ensures us that so that they are providing us this envelope protein in a very steady amount. Why? Because if there is no constitutive promoter, and if it is a tightly controlled promoter, and if the expression of this gene VSVG is expressed at a particular time point inside the host cell, it will start increasing the product of envelope protein inside the cell. Now this ENV protein can be toxic. It is toxic for our cell, as we all know that. Now if the concentration of ENV protein is getting higher and higher inside the host cell, it can make problem for the host cell. So for minimizing this, we supply this ENV protein in a very minute amount over time using this RSV constitutive promoter. Okay, now let us look at the process. So once we know that all these three different type of plasmids, and obviously we must have our plasmid carrying our desired gene. So what we will take here the generate the P lentivirus expression construct. So we take all of them and take the construct, and we have the package mix, and we'll transfect all of these plastids. Co-transfection is done in 293T cell line, 293T cell line again, which is a HEC 293T. It is also human embryonic kidney cell, and we co-transfect this cell with all these different types of plasmids. Then what we'll do, we harvest these cells and we take out these plasmids, and we again take this and put the mammalian cell line of interest. Then what we can provide, we can produce the expression and response depending upon our results. Okay, so this is the procedure of how we can do. Simply we take the vectors with the plasmids, which are 4 different plasmids. One is carrying our desired gene, and 3 of them, one is carrying the envelope protein, one is carrying the gag and pol, which is the structural protein, another one is called the transfer genes. And then we provide this inside the vectors. Sometimes the transfer gene can also have our own gene of interest. It can happen if we transfer 3 or 4 plasmid vectors here inside this 293T cell, which is a co-transfection machinery. Then we take out this plasmid from there, because after some times we get more and more plasmids inside, more and more viral, not actual plasmids, more and more viral particles coming out in this 293T cell. After the infection is conducted, we'll take out more and more to viral particle from there. So viral particle is being formed. Now this type of viral particle we want, so these are our desired particles, because these are disarmed and designed in such a way so that we can utilize them in the gene therapy purpose. So it purify it and can use it for the mammalian cell target. Okay, so we'll give this viral particle to treat the different type of genetic disorders or different type of disorders that are happening inside the mammalian cell line of interest that we get, and the viral supplement to the mammalian cell line is provided so that we can get our proteins. Okay, so that's how we can conduct this experiment. Now, what are the advantages or key features? The key feature is again the deletion of the U3 regions of 3' LTR to produce a self-inactivating type of plasmid, which is new for third generation. And again, the number of genes are reduced to only 3: GAG, POL, and REV. Okay, and the expression of GAG, POL is REV dependent. We have seen it. And the molecular cross-talking because between the PLP1 and PLP2 is also important. And the VSV gene is provided, which is from the vesicular stomatitis virus, but the promoter before this VSV gene is provided from the RSV virus, which is a constitutive promoter, so that the less accumulation of ENV protein is done at a time inside the cell. Okay, and none of this particular plasmid is contained a packaging signal or psi. That means none of the HIV-1 structural genes are actually present in the packaged viral genome, and thus are never expressed in the transduced target cells. So no new replication commented virus are produced. What is produced simply the desired virus, which is having all of these three different type of plasmids. Okay, so that is the role, that is the importance of utilizing this viral vector, and we can utilize in future. But again, this particular segment or this particular total part of our medicine or total part of our research about this uh, gene therapy is completely under review, and we need to look for it several round of uh, case study and several round of treatment or treatments is required to finally conduct something or finally conclude something about it. Okay, so that's it, and I hope this video is helping you. Thank you.